Welcome friend. Today's topic is your precipitation and concentration of the nucleic acids, that is your DNA. This book is your Molecular Biology and Genomics by Gordon Mulhart. Okay, so another typical problem here is that you can see that the while isolating a DNA, the most basic thing that we used to do is use alcohol like your ethanol, butanol, etc. Because they have the lower dielectric constant than that of your water, so it's easily precipitated. And the second thing is that we precipitate those DNA by using salt solution like your sodium chloride, sodium acetate, etc. So this is the two basic common things that being used for precipitation of the DNA in the isolation process. But this salt solution has some negative impact, and this method of alcohol isolations it also has some its adverse effect on this DNA that we isolate. So let's go move on, check this out, what kind of things that we will see, and what are different types of methods that we can use to precipitate this DNA. So first one comes in your row is the alcohol precipitation method. So the dilute a nucleic acid solution with the monovalent or single charge saline and add alcohol to it, and it will comes to precipitate. I mean the DNA will be precipitate. So the basic thing is your salt. So what kind of salts that we can use, and what should be the concentration of those kind of salts should have. The first comes to your sodium acetate. So it's about 0.3 molar should be used in alcohol, and this is the pH in which range it should be used. Similarly, sodium chloride and ammonium acetate, lithium chloride, etc. also in this row. And these are some pros and cons, and also I have underlined, you can see it very clearly. Next thing comes in our row is your alcohol, because it has dielectric constant lower than that of your water molecules, helps to precipitate the DNA. So first comes in the row is ethanol. Well, ethanol is very easily eliminated because it's evaporated quickly, and 70% volume percent is being used, 2.5 fold increase in volume. And this is the ethanol. Isopropanol is more effective than ethanol in precipitation of nucleic acids, and it can be removed less easily than ethanol. DNA pellets should be reused with 10% ethanol, so it's not good to use isopropanol, though it has a very effective precipitation, because the later on process that we use to remove those alcohol from the DNA is another difficult process. But if we use isopropanol in case of rather using ethanols. Similarly, butanol that we not use, it has some specific purpose also for using. Then concentration of nucleic acids. So whereas the nucleic acid is high concentration, you can see like when we are extracting the DNA from a tissue sample, we cross it and extract the DNA, and the DNA is huge in quantity. So in this case, where we guess that our DNA in the sample is about more than this range, we can use this kind of technique. But in the case where we don't have that much amount of DNA, we should not do this alcohol salt precipitation method, because it will not be able to give that much visible amount of DNA. So in that case, where our concentration of DNA lies in between 1 to 250 nanogram per microliter, so it's advisable to refrigerate the DNA in saline alcohol mixture at about -20 degrees Celsius for 24 hours, and that will lead to activity of that yield about this much amount of significant amount of DNA. Then the centrifugation is the common process, where we get a thin layer where we can simply get our DNA bands precipitated. Salting out is another process. In this process, what exactly happens in normal things? What happens exactly when there's no salts in solution, any chlorine or any compound like anything that is soluble in water and also surrounded by water molecules. But when there is excess amount of salts is there, then the salts will consume that water molecules surrounding to itself, and in that case the DNA or any kind of substances will not able to get able to surrounded by that much amount of water molecules, so eventually it gets precipitated to the surface, and that this is what we call the salting out. Then the phenol chloroform extraction method is used. Well, basically the phenol is being very highly toxic, and for that we use phenol, and we also use the chloroform substances because the phenol is not stable in that simple form. We have to use chloroform also. This basic reason that we should use this solution is to simply denature the protein molecules in our sample, so that there will be no protein in our samples left or associated with DNA. Also, we use protein escape, which kills this DNA enzyme, which is a protein also. And this is the